Hello, this is Marla, Mariana, and Marifer, and today we're going to start talking about DNA structure.
DNA stands for deoxyribonucleic acid. Let's take a closer look at the DNA. The structure of DNA are two strands, which are twisted, forming a double helix. In a closer look, each base is connected to a sugar, deoxyribose, and that sugar is connected to a phosphate. Thymine and adenine are bonded. They have two hydrogen bonds, keeping them together. Guanine and cytosine are bonded too, but they have three hydrogen bonds. Chargaff's rules explains this past thing. In the DNA on each organism, the amount of adenine is similar to the amount of thymine, same with guanine and cytosine. An easy way to remember this is apples in the tree and cars in the garage. Up next is DNA replication.
A little pun. What's better than one DNA? Two. First, the helicase separates the two strands of DNA, single strand binding protein, the SSB protein, and keeps the two strands from joining back together. Then the primase makes a path, called primer, for the next enzyme to do its job. DNA has two strands, the leading and the lagging. The DNA polymerase connects the bases with the ones in the two strands. He is lazy and doesn't do his job properly in the lagging strand. He leaves Okazaki fragments. The DNA ligase fills up these spaces. That leaves us in the end with two identical DNA which contains a new and an original strand. This is called semi-conservative DNA replication. Thanks for learning with us. The second step for central dogma is the RNA transcription. This is after the DNA replication.
DNA and RNA are almost the same. The difference between them is that DNA has two strands and RNA just one. Another of the differences is that DNA has thymine and RNA, instead of thymine, has uracil. They also have different sugars. DNA has deoxyribose and RNA has ribose plus one oxygen atom.
There are three types of RNA: messenger, ribosomal, and transfer.
DNA strand, the RNA polymerase makes a copy of one of the strands based on the bases in there and makes one single strand based on the one that the RNA polymerase. All this process happens in the nucleus of the cell. So, when the RNA polymerase makes the other strand of RNA, all of these go to the ribosome and the cell outside of the nucleus.
Ribosome is like a factory where the strand of mRNA goes through. This here is a tRNA. It carries bases and amino acids and works in 3, which are called codons. They match the bases they carry with the ones on the RNA strand.
The next topic is gene expression.
This little guy in here is a promoter. It guides the RNA polymerase, which is in charge of making mRNA.
The operon is mostly found in prokaryotes and in the roundworms. The promoter and the operator are inside of the operon.
The promoter is a DNA segment that allows a gene to be transcribed. Another segment is the operator. Its function is to indicate the gene when to turn off and on.
It blocks transcription from occurring when interacting with proteins.
What's an operon?
Well, the operon is another region of DNA which includes the promoter and the operator. It also includes one or more structural genes.
Where do we find the operons?
Well, as mentioned before, they are found in the prokaryotes and the roundworms.
The lac operon is one of the earliest examples of gene regulation discovered in bacteria. Gene expression starts with a very important character, RNA polymerase. Its main goal is to create mRNA.
Problem: RNA polymerase doesn't know where to start. This one is the other side. Solution: it's safer. It's hero, the promoter. This guy shows the RNA polymerase where to start.
Next to the promoter, we have the operator. Its job is to regulate gene expression by repressing it. This guy over here is called a repressor. Main goal: block RNA polymerase. Solution: lactose. Main goal: help RNA polymerase by attaching to the repressor. Congratulations, you have reached your goal. Prize: mRNA.
Let's take a closer look to the mRNA. In the pre-mRNA, we have introns and exons. Some introns are non-coding regions of DNA. They are rubbish. They'll be recycled later.
The survivors, the exons, gather together and form its own chain. This one will be transformed into proteins. Yay! The reason why they aren't thrown to the trash is because they are the coding region.